First, you don't see me because we don't want this video stream to be split into two ones, so it would be a worse quality. Can you make a little bit more light on your face? Can you put a
Okay.
little bit more light on your face?
Let me
Yeah?
change it for better light.
Oh, now it's perfect. Now it's perfect. Thank
Yeah.
you. So, may I ask you, what was the idea to, with this Daniel Ririo, I have read your articles, to take specific genes which are responsible for products sensitive to different pollutants? How the idea came to you?
Could you say again which gene you want to know?
How this idea to take these genes out of Daniel Ririo, special genes, has come to you?
What's the idea? Uh, so the system we engineer that we use uh, green fluorescent protein gene, which show fluorescent color. So in order to make the system working, we have to put a gene promoter in front of the gene to control the gene expression. So this gene promoter is the inducible promoter, and the gene can be induced by certain chemicals. So we find some gene promoter which can respond to certain chemical. When this chemical present, this will turn on the color gene, and the fish will show the fluorescent color. That's how this system working.
So this is a whole fish, or this is a genetically engineered fish, or this is a fish, or this is a subcellular system.
This is the fish, the whole fish, the live fish.
And you can engineer this fish. Which chemicals, which pollutants could be detected by this system?
Right now we have three types of fish, and one would detect the estrogen, which is a female hormone, and some similar chemicals. The second one is heavy metals, and which including several heavy metals. The third one is to have this certain category man-made chemical, which is called xenobiotics. This is mainly to restricted to a category chemical called TCDD or dioxin. So that's the three types of chemical we can detect so far.
What is the perspective? How can you use it in industrial scale, in cleaning stations, or in this cleaning stations, or this waste recycling industry? Could it be put into industrial scale, and how to do it?
Right now, still in the laboratory scale, and we just using this fish to demonstrate the concept. So the fish can be engineered to detect the chemical contaminant. So far, has not been really had the real applications yet.
So it's kind of startup.
Yes, it's a startup. And one thing that because this is a transgenic organism and a GMO, there's a lot of regulation. So we cannot use it in the wild. And so anything we want to detect, all we can do is to get water to bring to the laboratory, then to detect in the laboratory.
What is the advantage of this system compared to old ones? For example, before people used algae or mushrooms or daphnias to uh, mm, detect if the water is bad or good, and if the quality of recycling is good or bad? What is the advantage of your methods?
Okay, this in the fish system. Fish considered a high vertebrate organism, which share a lot of similarity to the human. Anything response in fish would be very likely to predict the health effect of human being. So that's the major benefit. Secondly, because of the system we engineer, we can detect the color in live fish easily, and so we don't need to sacrifice the fish for detection. And so the fish can be reused in the online system, and it can be repeatedly reused. And so that's a non-invasive method. Uh, I think that there was two major benefits to using this system.
To which pollutants can you potentially make these sensitive promoters? To which range? I don't know, because the range of pollutants and industrial pollutants is it's huge, like from small molecules like heavy metals to different toxins and so on.
So, so far the limitation is the inducible gene promoter, and the transgenic technology already been well developed. So it depends on whether we will have a available promoter to sense some chemicals. And so far only limited number of inducible promoter known. So they still need a tremendous research work and in order to find or identify more gene promoters. So the gene promoter basically like a switch for the light. If we have the promoter, we can link with the gene, then we can control the gene expression. So that's the main issue we needed to solve.
And there in Singapore, where you live, is very tough regulations on the quality of industrial waste or secondary water. How it is controlled now? How it is? What is our bioindicators which are used now, or this is a chemical control?
Right now, we haven't already put in the real use for this industrial waste, and we do have this wastewater reclamation project here. And so far, all the tests is just in the small scale in the laboratory. And initial the idea we engineer such a fish, we can develop a online monitoring fish for reservoir water. But we made some preliminary tests. The reservoir water so clean, so all fish basically at no use because everything would be negative.
This method so is more sensitive than chemical, or just the observation of the fish behavior in this secondary water. Yes, is much more sensitive.
Uh, I don't think our system would be more sensitive than the chemical method. And but the difference that our system is a biomonitor system can indicate the biological response. The chemical method could be more sensitive, but the problem that you have to know what's in the water, then you detect using the chemical method. If you don't know exactly what's in the water, and you may not be able to detect by chemical method easily. And in the biological system, and initially we don't care about what chemical in the water, but we will look at the biological response. In other words, if there's no biological response, maybe we don't have to worry about it because of the even there's such a chemical present in water, and it might be at a very minimum level, which will not cause any biological consequence. So there's a different system, and I would say this biological system and the chemical method would be complementary for the contaminant monitoring.
And how long does it take from the first contact of this genetically modified fish with the um, ligand, with the substance and the genetic response? How is it
Uh,
fast, and can you measure it quantitatively, the response?
yes, and we did some tests, and I think the response will require at least 6 hours because of from this chemical interaction to the gene expression to visible color, it will require at least 6 hours in the some of the transgenic line. In other transgenic line, it could take even longer. But usually the signal, fluorescent signal, will become stronger with a longer time exposure.
And can you measure the quantity, not the quality, but the quantity of the pollutants using these methods, genetical methods?
Yes, that's a dosage dependent response. If the concentration of such contaminant is high in the water, and we will see the higher fluorescent signal.
Thank you very much. Thank you. Thank you. Thanks a lot.
Okay, yeah, thank you.
Thank you.
Thank you.
Just if this is recorded, we just check it. Yes, thank you. Thank you very much.